Hello, and welcome to part two of Arcadia University's BI327 Histology lecture series on connective tissues. In part two, we're going to be taking a look at the connective tissue fibers. Again, part of the materials that are making up the extracellular matrix. In the previous lecture, we talked about the ground substance, which is going to be drawing water into the matrix. Now we're going to be taking a look at the fibers. And they're going to be generally three basic categories of fibers found within the majority of the connective tissues. We're going to have collagen fibers. Collagen fibers are going to be resistant to stretch. They're going to have a lot of tensile strength. They're going to be very, very strong. And basically, it's estimated that weight for weight, they're stronger than steel. So basically, you can tug on these collagen fibers and they're not going to stretch. They're going to have a lot of strength associated with them. In contrast, we're going to have some elastic fibers, another category of fibers within the connective tissue. And the elastic fibers are almost like little biological rubber bands. They have a lot of resiliency, but they're capable of stretching. They can stretch up to 150% of their length and then return or recoil back to their original shape, their original size. And so collagen, resistant to stretch, elastic fibers can stretch and recoil without damage to the structure. And then the final type of fiber we're going to look at within this series of lectures is going to be the reticular fibers. And the reticular fibers are a specialized collagen fiber that's involved with forming a very fine supported network. It almost forms like a jungle gym-like structure, three-dimensional structure for a region, for cells to migrate through. And we'll talk about some specific examples of that as we go through this course. Now, the first one we're going to look at is going to be collagen. Collagen is the most abundant protein within the body. Lots and lots of connective tissue, lots and lots of need for collagen within the body. Now, collagen is a triple helix, which means that it's essentially three amino acid chains that come together and coil together into an individual collagen molecule. Now, every third amino acid is a glycine. If you remember your biochemistry, glycine is the simplest of the amino acids. It has a hydrogen for that residual group, that R group. And so it's going to be the smallest of the amino acids. And that's important because it's very, very small, which means it can not take up a lot of space and that this collagen molecule can twist around very, very tightly. There's going to be a lot of hydroxyproline and hydroxylysine within the collagen. That's an identifying characteristic of a collagen molecule. But it basically is going to be this triple helix, this three amino acid sequence chains that are twisted around with one another. So they're going to be very, very strong because you got like three components coming together and giving it kind of that flexible, like a rope, but lots of tensile strength. So it's almost like a braided rope. You can twist the rope around, you can move it in different directions, but when you tug on the rope, it's not going to stretch. So it's very, very strong. We think about this, collagen is a classic protein. Lots of protein structure associated with it, lots of amino acids. So it's going to stain very pink with eosin, and so connective tissues are in general going to stain pink, at least the basic connective tissues. With some of those specialized stains we talked about in one of the earlier lectures, like the trichrome stain, it specifically stains the collagen kind of a bluish green appearance. Now, if we take a look at the collagens, there's actually 5 different types that we take a look at. Type 1 collagen is going to be the most abundant and most widespread. And if we take a look at that in the diagram on the bottom portion of the slide, shows you type 1 collagen becoming organized. So you've got the individual little chains coming together into the procollagen and the tropocollagen, but basically forming that triple helix that we talked about a couple slides ago. But then those triple helixes come together and interact in the formation of what are referred to as fibrils, where basically lots and lots of these individual little collagen molecules coming together, and it's going to be in a repeated pattern. They're going to be twisted around one another, and they're going to form fibrils and ultimately fibers and fiber bundles. And it's going to have a banded or striped appearance because this repeated staggered pattern of the collagen molecules coming together. But what it means then is that the type 1 collagen is almost like a braided rope. If you had the time to take a tow rope out, and you can see it's all twisted around, and it's all braided around, but you start to dissect it out, if you start to pull at the little threads within it, you're going to see that each thread is going to be relatively short in relationship to the entire length of this this tow rope. But it's the fact that they're all together, they're twisted around, they're braided together, they're intertwined with one another, means that these relatively small molecules, or pieces of thread in a braided rope, but these small collagen molecules are going to be interacting with lots and lots of other collagen molecules, forming very, very strong fibrils and fibers. So very, very strong structures. So we can find light type 1 collagen in things like tendons, ligaments. Later on, we're going to see it in bone, woven within the dermis, deeper regions of the skin. We can find it in organ capsules. We can find it in loose connective tissues. Lots and lots of regions of the body where it's giving its strength. Type 2 collagen. Type 1 collagen is the most abundant. We talked about that. Type 2 collagen doesn't form those heavy fibers. It only forms these very thin fibrils. So it doesn't have the kind of clunky structure that the type 1 collagen has. Type 2 collagen, these thin fibrils of collagen, are only going to be found in the cartilage matrix. And we'll talk about that in one of the lectures coming up. Type 3 collagen is going to be very, very similar to type 1 and is found associated with type 1 collagen, but it's glycosylated. And so basically, it's going to be interacting with type 1, but in general, it's going to form thinner fibrils as opposed to the thick fiber bundles of the type 1 collagen. And you can find type 3 kind of woven around type 1 in narrower areas and surrounding and supporting flexible tissues like smooth muscle cells, nerve fibers, fat cells, adipocytes, things like that. Type 4 collagen, again, a variation of this collagen molecule, is going to be found in the basal lamina. And we've actually seen that on previous slides, but we didn't emphasize it. But it's essentially going to be a strong protein, a fiber-like protein, but it doesn't form the fibrils and fibers at the gross level, kind of the larger multi-protein levels coming together, like we see with the other types of collagen, but it's there for strength. It's going to be intertwined and interwoven, forming a good, strong supporting meshwork within the basal lamina. And then finally, we have type 5 collagen, which is found rarely within the body. It's within placental basement membranes, it's in some of the blood vessels, and in small amounts elsewhere. Now, if we take a look at this, the collagen fibers are going to be very important. If we take a look at an inherited defect in the development of the collagen fibers, like Ehlers-Danlos syndrome, what we can see is that we have defective collagen fibril formation, which basically means we have a very dramatic reduction in the strength of the collagen molecule. And so what we can see are things like hyperelasticity of the skin. If you pull on your skin, it's going to be relatively resistant. If you have a breakdown or disruption of the strength of the collagen, you're going to have a lot more stretch there, a lot of elasticity associated with it. You can have joint dislocation in Ehlers-Danlos syndrome because of the weakness of the collagen molecules, because you've got the tendons and the ligaments, which basically are weaker and they're not holding everything in place. And again, looking at this as an important emphasis on what the collagen is doing within the body, holding the structures together. Now, moving from the collagen, which are very, very strong, almost like steel molecules, flexible but strong like steel, to the elastic fibers. The elastic fibers are actually made of an elastic protein, an amorphous protein called elastin, and some protein microfibrils, other things that are going to be interacting with it. Relatively narrow, about 0.1 to 10 micrometers in diameter. And they're basically like rubber bands. Now, in general, elastic fibers are going to stain very poorly with standard ionic dyes. So they're going to stain pale with hematoxylin and eosin. You're not going to see them because they're not going to take up any of the stain. But there are a couple different examples of specialized stains that we can use to look at elastic fibers. The first is nicely named elastic fiber stain, which gives it kind of a bluish black or reddish purple staining, depending upon the characteristics of the stain and the amount of elastic fibers that are present. We'd also look at an aldehyde fusion stain where they're going to be staining these elastic fibers purple. But again, the characteristics of these elastic fibers is they're like rubber bands. They can stretch and recoil without damage to them and damage to the surrounding tissues. So they can be very, very pliable, very, very elastic, and like rubber bands, they can stretch and recoil without damage. And so we could expect to find elastic fibers in tissues that need to be able to stretch and expand in return without damage. And so lots of elastic fibers, especially within the larger arteries, which when the heart pumps, a large bolus of blood is going to go through, stretch the artery out, and then when the heart is relaxed and refilling, the artery walls are going to come back to their normal size. They're going to recoil. Interalveolar septa, bronchi, bronchioles of the lungs, all of which are going to expand and recoil as we're going through that inhalation and exhalation process. Vocal ligaments are also going to have some elastic fibers associated with them. We take a look at Marfan syndrome. Essentially, we're looking at another example of an inherited disorder. This has both a pathology of both the collagen and the elastic fibers, can contribute to a whole range of effects. But basically, it's associated with the connective tissue being too lax, too elastic. So it basically expands out and doesn't recoil real easily. And so it contributes to a lot of cardiovascular defects. Things like the periosteum, the structure around the outside of the bone, is too elastic, and so we get a lot of skeletal defects because we need the forces on the bone to direct the deposition of bone, to direct the strength of the bone structure. And we don't have that if we don't have the support and the structure in these connective tissues. The final type of fibers we're going to look at are going to be reticular fibers. And these are going to be similar to the collagen fibers, but much, much thinner, on the order of about 0.1 to 1.5 micrometers. These are going to be highly glycosylated, so lots of sugar groups associated with them. They're going to be some type 3 collagen, lots of glycoproteins associated with them. But they're going to form essentially a relatively delicate three-dimensional matrix. And so you can think about it almost as like a jungle gym-like structure. And so they're going to form a lot of open spaces within a region of an organ, but it's going to allow motile cells to essentially migrate through the region. So like kids playing on a jungle gym, the cells are going to move through this reticular connective tissue and be able to migrate through the region. So we can see this within hematopoietic tissues, the tissues involved with blood cell formation, and some examples of lymphoid tissues, some of the immune system organs. Often difficult to see these reticular fibers without special stains. We can use like a silver salt stain, which will cause these to stain kind of a black appearance. And you can see almost this three-dimensional jungle gym when we take a look at the image on the right-hand side of the slide. And that finishes up our discussion of connective tissue fibers. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thanks, and we'll see you in part 3 of this lecture series.